In the interest of keeping us on schedule, I would like to introduce Dr. Feng from Clemson University. He's an associate professor of biophysics. His current research focuses on multi-scale modeling of biomolecules and molecular complexes. So I'll turn it over to Dr. Ding.
Okay, can you guys see the screen? Let's see.
Yes, and we can hear you well.
Okay, thanks. So first, let me thank RAMS for organizing such a great symposium and also all the amazing talks here. First, I would like to thank my previous speakers, including Sam and Lucia, talking about amyloid IAPP aggregation and also the endogenous inhibition. So this actually what I'm going to talk about. So actually I will skip first few slides. Okay, as I already mentioned, so IAPP aggregated very fast. It's one of the most amyloidogenic proteins in vitro, at least we tested. But inside the granule, it's like millimolar, so it should, why didn't aggregate, right? So hypothesis is you have other co-aggregate, like a co-component, co-localizing component, including insulin, actually a pre-proline C-peptide, which is the byproduct for making insulin, and zinc, and definitely low pH, because pH is around 5.5. So, so that's actually also the working hypothesis we have been trying to study in my lab. So my lab is computational lab, so we're trying to look at the structures, the interactions, and maybe trying to gain some insight. And this problem has also kind of been asked quite early by Westmark and also by late Steiner, published in Phipps later back in 1996. So, and also there are, when I started to ask these questions, literature suggests that, so here I will focus on zinc. So the people have using association study found that there are some association between this zinc transporter 8, some mutations associated with increased T2B risk. For example, this particular mutation, W325R, it's a mutant suggests to reduce the activity of the zinc transporter, which means the mutation will reduce the zinc inside the granule, and they have a slightly elevated T2D risk. But then there also, when I started, I read this at that time back in, I think 2013. I think in Nature Genetics, they found that there are loss of function mutation of this zinc 8 transporter, which is specifically expressed in the beta cell. That suggests that loss of function of this mutation causing basically loss of this transporter protects against type 2 diabetes. So this actually create a puzzle. So why some data suggests that reduced zinc promotes type 2 diabetes, but then complete loss of zinc transporter actually protect. So apparently, definitely, there's a lot of interesting questions that can be asked. So this is a kind of 3 topics I would like to talk about. So first is kind of trying to address this question. So first is asking whether zinc-dependent insulin oligomerization may affect IAPP aggregation. And second, I'm going to talk about whether direct zinc binding would, what's the effect on zinc aggregation, which actually has been discussed in previous talk. So, but anyway, I will entertain everybody in the audience, try to see what we can learn from maybe molecular modeling. And third one is, how about maybe it's a cooperative effect. Maybe it's an effect instead of one component, but a mixture of different components may act on endogenous inhibition of IAPP. Anyway, before we move, I just want to quickly advocate my approach, which we used in our lab. It's called discrete molecular dynamics. So it's a special flavor of molecular dynamics. Basically, we're still solving using the same physics. Instead of using Newton's law, F equals ma to integrate the equation of motion, we basically approximate the interaction by, let's say, step functions. This is just simplest ever version, like a one-step attractive well, but it captures the main feature of, let's say, van der Waals interaction. You have no interaction far away, some weak attractions when they're close, and they cannot go further. But you can increase the details to capture more details if needed. But anyway, so by doing that, we don't solve integrate the differential equation, but rather using conservation laws, of like energy conservation, momentum conservation, angular momentum conservation to solve the same, we call it ballistic equations. Just a fun fact, the first ever MD simulation is by this Alder and Wainwright back in 1957. That's a hard sphere model, which is we can call it a DMD. Anyway, so I will not go in further details, but over the year have been developing this method to try to study biomolecules. So let's first look at the first problem. This is actually just some I took for, actually I should have added the reference. So basically what they show that if you don't have, if you have a zinc, so this is with the regular like image of EM image of the beta cell. And you can see that inside there are some dark image indicating, sorry, this granule have this dark center, which actually reflect the crystals of insulin. But then if you don't have a zinc transporter, basically just deleting the transporter, you see more sprayed kind of image without seeing any forming any insulin crystal. So another fact is actually we have in our pancreas beta cells, we actually have a lot of, because of the zinc is believed to facilitate insulin crystal. Actually, they form mini crystal in our beta cell granules. So then at that time we were thinking, okay, how about maybe we looked in the literature, and there are some studies showing that zinc looks like indeed facilitated the crystallization of insulin. Actually, it forms in the presence of zinc, it forms a insulin hexamer. And the insulin hexamer is actually not very soluble. They will aggregate, they will crystallize. But in without insulin, without the zinc, it believes that actually they in equilibrium of monomer and dimer equilibrium. So this is a monomer dimer equilibrium, and then in the presence of zinc, they will actually move the equilibrium first further to the hexamer, and this is regulated by this called zinc transporter. And so the question is, maybe the oligomer or equilibrium might have an impact on IAPP aggregation. So the first we looked into the binding between insulin and IAPP. So this is just showing the dimerization we did in the simulation of two insulins, and we found that they do the amyloidogenic region, which is around residues 22 to 27, or in other words, people call NF-gal, that actually do pair with each other, forming a beta sheet. And also the first N-terminal helices are relatively stable, looks like. So when we simulated binding with insulin monomer, we found that the B chain of the insulin monomer actually forming a beta sheet-like structure with the amyloidogenic region of the amylin. So, and interestingly, this is B chain actually in the dimer actually will be buried, and also further when all this in the hexamer, all those interface will be buried. So what basically, okay, this is just heat contact map. But anyway, so then based on this, we came up with a kind of a simple model at that time. So basically, we'll say that, okay, IAPP alone, definitely, they probably initially form dimer, and then the amyloidogenic region will form parallel beta sheet, and then maybe once they come close, the N-terminal helices will unfold and further grow into fibrils. And in the presence of the zinc insulin monomer and dimer, basically monomer will compete for the amyloidogenic region and prevent the basically what happened. This finding first, you could slow down the aggregation of self-association of amylin, and also you could serve as a capping. Basically, when the amylin start to grow, the fibrillage, this insulin could cap on this end and prevent its further growth. And so basically it suggests that if you don't have zinc, so all this insulin crystals don't form crystal, but rather stay as monomer and dimer in equilibrium, and they will be very efficiently inhibiting IAPP aggregation. So that's the kind of first study we did on this topic. And then basically, we also looked into direct binding of zinc with IAPP aggregation. In this earlier work by Steiner et al., and they found that when they add zinc chloride to this is a pretty high concentration, it's 250 micromolar of solution of IAPP. He found that initially with the increase of the zinc salt, he sees increased aggregation, but then eventually it will decrease. So that was, although if you look at the relative molar concentration, this promotion of aggregation happened at the substoichiometry of the zinc. And also this looks like a slide on this, compared to what we heard in the last talk, which is by Ram's group. They see that when you add increased zinc, we see reduce of aggregation, right? However, I think the major difference here is relative ratio, because in this work, I think all this zinc is probably above the equal or higher molar concentration compared to IAPP. So first, as we heard in the first section, Birgit talk about modeling of ion is actually not trivial. She mentioned like a bonded model, a non-bonded model. So at the first, what we did model is look at a bonded model also. So based on the work by Rams et al., they show that zinc do bind to histidine 18. And also, although there are evidence showing that they might bind other residues, but it looks like histidine would be the strongest affinity, but strongest primary binding site. They might have additional binding site, but definitely will be weaker. So we first is using modeling called a bonded model, but we think if it's to mimicking the stoichiometry, different molecular ratio. So what we can think is zinc coordinating, like bind to 1 IAPP, 2 IAPP, or so on and so forth. So maximum maybe hexamer up to 6 coordinations. So anyway, so indeed, basically we just model each of these oligomers, zinc coordinated oligomers separately, and try to understand how when they form such a complex, so what will be change in terms of inter-IAPP interaction. This is just DMD simulations. Although here the unit is a nanosecond because it's implicit solvent, so we believe it's actually mimics up to longer time scales. But anyway, so we found that although there are some zigzags, which is because averaging over many independent simulations, and then we try to fit those looks rather noisy curve of number of inter-IAPP hydrogen bond formation. And we found some quite interesting results. So this is okay, so this is to IAPP control, and look at the number of hydrogen contact between the amyloidogenic region. And the second one is the number of interchain hydrogen bonds. So we see similar behavior, kind of trend is that with so moving from left to right is this is without zinc. This is 2-dimer, but then this is a hexamer of IAPP coordinated by zinc. And as this mimicking basically increase of the zinc relative zinc protein coordinate ratio, we can see around this tetramer, we found that they have a highest contact formed between the amyloidogenic regions of IAPP and also number of hydrogen bond between different chains. So so basically suggests that when you have substoichiometry of ions, ion may facilitate oligomer formation. Inside those oligomers, especially for those highly compact oligomers, as it increase, so basically effect is kind of, you can think it's a local high local concentration of IAPP. And then so they may facilitate the nucleation of initial fibrous seed. So so that's kind of our model. Also, when you have 1-to-1, let's say each IAPP bind to one, so this is actually each IAPP bind to one zinc. So what happened is now this whole complex will be will be more positive charged because IAPP itself is +2 charged. Binding to a +2 zinc, what happened is the net charge of this actually increased. So as a result, the probability of them to forming aggregate will be reduced. And also, if we also did this, is actually we protonate histidine 18. So basically mimicking, but make histidine 18 become positive charged, which would happen at the low pH, and that also reduced the aggregation. So anyway, so this is just a cartoon representation. This x-axis is increased zinc chloride concentration. This will be aggregate. So initially, without any zinc, they will itself will aggregate and form beta sheet and form nucleate the seed, and then seed will grow and form the fibrils. So as you with the low substoichiometry of zinc, they may promote oligomers, and those oligomers might facilitate or decrease lag time and promote aggregation. But then as it ratio increases, this zinc binding to the histidine effectively basically creating increased electrostatic repulsion that will inhibit aggregation. Further, if you keeps adding it, because let's say you have the millimolars, like the tens of millimolars, what happens is, talking about in vitro experiments. So what happened, you will have also a lot of ions, will have like chloride. So the screening effect will take effect. Actually, we're causing a kind of slight increase observation of increased aggregation. So anyway, this is a very simple model based on, it's called bonded modeling of zinc binding. Okay, so so this we also had collaborated with our collaborator at Clemson. So they also did different relative ratio. We do see an initial decrease of. We see initial increase of lag time, eventually also decrease. So basically, kind of consistent with our prediction. So the last I will talk about is the cooperative effect of zinc C-peptide on IAPP aggregation. So we also know that they have a huge amount of C-peptide, which is a truncated sequence when making insulin. So what happened is C-peptide has a lot of, it's highly negatively charged. It has actually 5 acidic residues. And then this is just a sequence of the insulin. You have 2 positive charged, so it doesn't have any negative charged amino acid. Although here, this is histidine 18, which is shown to be have a stronger binding with zinc. And for the C-peptide, and there are also some AMR studies showing that suggesting that zinc do bind those negatively charged amino acid and causing compaction of the C-peptide. Okay, so in this here we start to introducing this called start to learn how to do non-bonded models. We basically here we don't constrain them. We just also in this model we actually also introduce a directionality of zinc binding, especially for example when zinc bind with the histidines will also tend to be coplanar and with specific angular and distance dependent. So anyway, so I will not go to details about the model itself. But anyway, so here what we did is if we add zinc together with one IAPP, one C-peptide in box simulation. But for now, this is averaged binding probability of zinc with IAPP and C-peptide in this simulation box, averaged over 100 independent simulations. And we found that zinc actually interestingly bind C-peptide faster and with high probability, and with IAPP actually is relatively slow and also with lower probability. The reason is very simple, because ion is those long-range electrostatic interaction will facilitate its rapid binding with C-peptide, but with IAPP is actually slower. And we also find that binding with C-peptide is actually clustered in the N-terminal, because you have 3 negatively charged amino acid in the vicinity. So it turns out that this will have a strong binding with the zinc. And the one in the downstream near the C-terminal will be lower probability, although it can bind, but it's lower probability. And this is showing that at the end of simulation, in forming this heterodimer with zinc coordinating one histidine of the IAPP and bind to the N-terminal of the C-peptide, but also have some dynamic binding with for example, E27 and E11. And also when we examined the heterodimer, we found that zinc actually looks like, first we found that zinc in many of time is still exposed and also not saturated, because we, based on the analysis of the crystal structures containing zinc, we found that zinc actually can coordinate up to 6 chemical moiety, like either nitrogen or oxygens. So then we found that we actually modeled the heterotrimer, basically have one zinc, one CPP, but with one C-peptide, but 2 IAPP. And we showed that the binding is actually, first of all, will saturate the zinc. And also we found that in this heterodimer, the IAPP become more helical, and especially in the C-terminal, the amyloidogenic region, which is 22 to 29, 27 or 29. Originally, it's in this heterodimer is the medial helical, but when you have the heterotrimer, the IAPP become highly helical. So this we believe that will stabilize the IAPP and from it prevent it from further aggregation. And this is just a testing, adding another incoming free IAPP. We show that actually because of this IAPP now is in this complex is highly helical, and also it's the amyloidogenic region is protected, that it does not form interchain hydrogen bond with the incoming one. And this is in collaboration with Puchun Key's lab at Monash University. Now he moved to Guangzhou. So basically, the main conclusion is when we fix the IAPP concentration, but add increase different ratios of zinc and C-peptide, we found that with this roughly, although this ratio here is 2.7 to 1, is like more than 2 C-peptide. we do see the reduced in the amyloid aggregation, and also for the measuring the final fibers is actually it's very short. And also we found that in this mixture we do have let's see, this is the analysis of fibers. Yeah, so basically showing here, you actually see that you have a smaller, less aggregate, and also the final aggregate is actually it's relatively short comparing to the controls. Yeah, and also those are, I think it's in situ using ex vivo experiment showing that one thing actually I want to mention that if you add a C-peptide with IPP, we found that it promoted aggregation. But interestingly, we found that, so this is just comparing with adding IPP to the cells. If you add a C-peptide, it promotes aggregation. It turns out that also the promoting or accelerating aggregation actually reduce the toxicity, which also suggests that the mature fibers is probably not very toxic. Yeah, also here, when we, this is together with IPP and C-peptide, we show a much lower cell death. Okay, so here I just want to make a final summary. So we believe that zinc play a very complex role in the kind of endogenous inhibition of ID aggregation inside the granule, because we know that in healthy individual, they don't aggregate. So zinc play a very important role, but complex. And promoting those endogenous inhibition might be a potential strategy to for prevention of type 2 diabetes. Here, just a cartoon kind of summarizing the results. So this is the effect of insulin. They may regulating the insulin oligomerization, and that could the more insulin soluble insulin might inhibit have a better inhibition of IPP aggregation. This is just illustrating how zinc might promoting oligomers if it's substoichiometry, but if it's high concentration, actually inhibit aggregation. And this is actually talking about co-aggregate with the C-peptide. Anyway, so I hope that I convinced you that DMD is also could be a very predictive tool to study aggregation. And also I would like to acknowledge like hard work of my former student, although Praveen, he's my former postdoc. And first two stories were done by Praveen. And Xinwei, who graduated a few years ago, now he's in biotech doing data scientist. Then Ye is a postdoc in my collaborative lab, did the experiment. And also Emily and Alexander from Monash, those are postdocs of Puchun and Thomas Davis at Monash. And also some ex vivo work is done by Esteban. So I thank also funding agencies from NSF, also NIH, also when I started the startup fund from the Clemson. So thank you. I'm very happy to answer your questions.
Okay, please put your questions in the Q&A section or the chat. Be happy to read those out. So this is from Anoop Oranguri. A question for Feng. Your last slide said C-peptide promotes oligomerization, but I could not follow the role of zinc there. So I think he's wanting some clarification.
Right. So when I add C-peptide alone, without adding any zinc, just a pure C-peptide mixed with IPP. So what happened is the mechanism is because the C-peptide is more negative charge. Actually, you can see it doesn't really have a positive charge amino acid, so it has a -5. And IPP, we know that is +2 charged typically, although if considering the N and C termini capping, but this is +2, this is -5. So what happened is each C-peptide actually combined due to electrostatic pairing can bring multiple IAPP together basically will promote the self-association of IAPP, and that's why it promoted aggregation or promote IAPP aggregation. So we found that if you add mixing IAPP with C-peptide alone with changing the concentration, we do find that under most cases IAPP do promote increase the fluorescent incubation time and also reduce the actually this is lag time, sorry. Basically promote the aggregation. You can see more fibril formation in the when you add C-peptide.
Thank you.